Dear colleague, dear friends, it's a great pleasure to welcome you to this new EHAT podcast, which will focus on gene therapy. As you know, EHAT podcasts are increasingly popular, and they also contribute largely to giving more visibility to the multiple activities of the EHAT Academy. So this one will focus on gene therapy, and I certainly invite you to discover the full program on our e-learning module. Our guest today is Professor Thierry VandenDriessche, expert in gene therapy. Thierry, it's a great pleasure to welcome you today.
The pleasure is mine, Cedric. Thank you for having me.
So Thierry, maybe you could introduce yourself.
Well, I am actually professor at the Vrije Universiteit Brussels in Belgium, and I'm running a gene therapy department that is fully dedicated to gene therapy. I've been working on gene therapy for almost 40 years. It is a journey that started with my master's thesis on gene therapy for cancer, that was, and I gradually moved into gene therapy for monogenic diseases, particularly haemophilia.
And how did you get so much interested in gene therapy?
Well, Cedric, thank you for the question. This goes way back. In fact, as a teenager, I got my hands on a book of J.D. Watson, who unraveled the structure of DNA, molecular biology of the gene, and I was hooked. So since then, I have never weaned on my interest in genes and gene therapy. But really, Cedric, what from a philosophical point of view, what triggered my interest in gene therapy for monogenetic diseases is the fact that a single point mutation in this jungle of 3 billion nucleotides that makes our human genome can make all the difference between life and death.
Yeah, this is true. So, well, as our listeners know, the purpose of this podcast is really to give a flavor of the full program with all the slides, the beautiful slides that you prepared that are now available on the EHAT Academy platform as an e-learning module, focusing on gene therapy. And in this module, you focused a lot on the limitations of gene therapy, the current limitations. So could you summarize quickly what are, in your view, the current major limitations?
Well, the current major limitations are, I believe, twofold. One, the significant interpatient variation. We don't have a handle over this. We cannot fully predict how the patient will respond, where they will end up in terms of their factor VIII or factor IX levels after gene therapy. But perhaps also the second most important limitation from a safety perspective is the fact that many patients do develop transaminitis, and we need to get a handle over that. And I believe there is a way forward in this.
And in terms of immune response against the viral vector, do you see that as a major limitation currently?
Well, the immune response against the AAV vector is an important limitation in the sense that we cannot re-administer the vector if the expression declines. And we have seen evidence from clinical trials that in some studies, there is a slow decline in expression, particularly in the AAV factor VIII studies. So if there is a need to re-administer the vector, that's not going to work because the person has in a way been vaccinated against AAV, will have antibodies against the AAV vector, and so cannot be effectively retreated. But there are ways around this, Cedric, and we can either do some capsid engineering to minimize the reactivity with the antibodies. That works to some extent. It's not black and white, you know, but you can at least reduce the immunoreactivity. A second way is to try to adapt the patient to the vector as if it were, and administer certain agents that minimize the immune response, immunomodulatory agents, certainly not unknown to the treaters of hemophilia gene therapy subjects, because corticosteroids have been out there for a while. Whether this will solve all the problems is very unlikely, but I do think that there are some really innovative approaches to that have been validated in preclinical models and that allow us to re-administer vectors, even if they, in the face of low titer antibodies against AAV.
Yeah, and I would like to emphasize that in your lecture, well, there is a complete full section which is dedicated to this very important area of research, which is focusing on how to improve AAV gene therapy. So you discussed already about, yes, the vector, the capsid, but if we now focus on the gene, what could be done at the gene level that could improve the success of gene therapy?
Well, Cedric, what we can do is we can learn from nature. In fact, thanks to the work of Paolo Simeoni, hyperactivating mutation in factor IX has been identified. And this is now widely known as the factor IX Padua because the patient was from Padua. And it's a hyperactivating mutation that results in a 5 to 10-fold increase in factor IX activity, which in that very patient triggered thrombophilia. So we set out many years ago with the research to test if we could use this Padua factor IX gene to improve the efficacy of gene therapy for hemophilia. And we showed that indeed, in the context of hemophilia gene therapy in preclinical animal models, we could then demonstrate a 5 to 10-fold increase in factor IX activity. And our work was published and then head-to-head with the work done by Valder Arruda as well. And this actually triggered a renaissance, if I may say, of factor IX gene therapy, because indeed it can allow us to obtain much higher activity levels in patients. Since then, this has been confirmed in patients and led to the first AAV factor IX gene therapy products for hemophilia B.
So this is a very important strategy, is to modify the gene, making it more efficient. And maybe, well, this is something I'm less familiar with, but do you think that the level of the promoter, anything could be done in the future to improve the efficacy and success of gene therapy?
Yes, absolutely. Thank you for the question, Cedric. In fact, there are 3 different factors that we can play with. We can play with the gene, as I just explained, for factor IX, for factor VIII as well, by the way. We can do very nice bioengineering on factor VIII. We can also work on the AAV capsid itself. And finally, we can work on the promoter. So it's like an intricate molecular ménage à trois, if you may, that we have to optimize. It's challenging, but it works. And we have actually worked a lot on promoter engineering. We've maximized the potency of promoters so that we can increase the levels 5 to 10-fold at least.
Yeah, so that really opens quite interesting perspective. And in your lecture, you had also a very important section that was dedicated to the potential of integrating the gene. So, well, my understanding is of current gene therapy is a little bit like adding a USB stick to your computer. So you had a piece of information, so it does not, go into your hard disk. But here, if I understand well, but you're the expert, when we think about integration technology here, I think it really means working on the hard disk. Is that correct?
I really like your metaphoric analogy here. It makes perfect sense indeed. The AAV vectors, they hang around in the nucleus and they don't really integrate, or at least only a fraction of those integrate, to be precise. So it's indeed like a USB stick. But what we really want to try to achieve here is to tamper with the hard drive. Remember, a single nucleotide can make all the difference between life and death. So how can we correct these genetic mutations in this jungle of 3 billion nucleotides? And there's actually, of course, 2 ways of doing that. One way is, well, we leave the mutation as it is, but we integrate a functional copy of the gene somewhere in the genome randomly. And that's what we and others have been doing with integrating viral vectors. But of course, it'd be a lot neater if we can, with nanomolecular precision really edit the very mutation that caused the disease. And then we enter the realm of gene editing. And that's looking for a needle in a haystack, Cedric. Can you imagine? You just have to find out where that mutation is. And with molecular Tipex, you can correct it and correct the disease. I think from a conceptual perspective, this has been truly amazing that we can do such a thing.
Yeah, and if I'm correct here, different viral vector are used. So these are lentivirus. And if I'm correct, you have been working on this for a long time. I noticed that you published a very important paper in PNAS in 1999. So how does this integration work here with this lentivirus? And also, I remember that you explained how it works in different animal models. Am I correct?
Yes, absolutely. I think in the very beginning, there was actually no firm proof that hemophilia A, in that case, could be corrected by gene therapy. It was an idea, it was a hypothesis. We needed experimental data to prove this. And I remember in the 1990s, there was a critical report that was issued by the then NIH director, Harold Varmus, saying, well, gene therapy is exciting, but guys, we need proof that it works. And so that inspired us also to go really delve deeper into the technology and really in an evidence-based manner show that gene therapy can work to cure hemophilia. So what we did in those days was to inject an integrating vector, an early generation retroviral vector, into newborn mice. So they were just 1-2 days old, a single shot or maybe 2 shots on 2 consecutive days. And then we saw that we had lifelong expression of our factor VIII. They essentially died of old age, Cedric. Many of these mice, they were old. And that's actually I believe the holy grail in gene therapy in general and gene therapy for hemophilia is we want the patients to live as long as possible and to die of old age, just like you and me, I hope, touch wood.
Yeah, and what you also discuss in your great presentation is the potential to target other cells, because we know that the hepatocytes are the cells that are mainly targeted, but you also showed that experimentally it's possible to target very important cells in terms of factor VIII production, which are the liver sinusoidal cells or even other cells. So could you elaborate a little bit on these strategies?
Yes, that's also a very important issue, Cedric, in the sense that historically we have been focusing on hepatocytes that normally express factor IX, but they do not normally express factor VIII. These are indeed the liver sinusoidal endothelial cells, but there's plenty of evidence out there by many groups, including ours, that you can express factor VIII in hepatocytes. But that's not the cell that is normally programmed to express factor VIII. And we could try to deliver factor VIII into the LSEC instead. We can again use lentiviral vectors in order to achieve that. Lentiviral vectors do transduce liver sinusoidal endothelial cells. We also showed this more than 20 years ago. And we can actually use endothelial targeted promoters to express factor VIII specifically in LSECs. And this actually works. The levels are not as high yet as what we can routinely achieve with lenti and hepatocytes, but we're getting there. I think with additional vector engineering, higher transduction efficiency with alternative envelopes and better promoters, we can eventually crank up the levels in LSECs. And that's really important because I strongly believe that if we force expression of factor VIII in a cell that normally doesn't express it, it may perhaps trigger other cellular responses, like a cellular stress response.
I agree. Well, time is running, and I would like to cover 1 or 2 additional topics before we close. Maybe a few words about these concerns that is shared by a large part of our community, which is the risk of genomic integration, and what could be done to mitigate this risk of oncogenicity when you use this kind of integrative approach. A few words about this?
Yes. Well, integration is a double-edged sword. You know, we and others have shown if you have if you have genomic integration, the gene is not going to pop out again. It's there forever. And that's a good thing in terms of lifelong expression. of our clotting factors. But we can't really control where these lentivectors or retrovectors integrate. We can't control that for now. There's ways to do this bit by bit. We're getting there, but it's not yet ready for prime time. It's not yet ready for clinical applications. I'm talking about site-specific integrating lenti, that is. But really, even with the lentivector, if we look with a panoramic view at all the lentiviral-based gene therapy trials for other diseases beyond hemophilia, there's really no evidence that the current generation lenti results in insertional oncogenesis. We have to look at the data. That risk is very low. And the reason is, Cedric, is that the devil is in the details. If you carefully design your vector, you can minimize the risk of insertional oncogenesis. And in my presentation, I show some data, how we can validate this. What can we do to improve the safety? To make an analogy with a car, you have a brake and you have the gas pedal, Let's forget about the clutch for a second. But you want to have control. You don't want to drive your car just with the gas pedal in. You need to be able to release that. And the vectors that we have now, they have that modality built into them. They're much safer. So I think it's incorrect to assume that if you have an integrating vector system, it's inevitably going to result in insertional oncogenesis. It's much more nuanced than that. But I think at the end of the day, we now have paradigms based on CRISPR, where we can really modify the genome in a targeted way, either in the factor VIII or factor IX gene itself, or in a region of the genome that we call a safe harbor, far away from any dangerous gene like an oncogene. And there's so much evidence out there now that gene editing also works to cure hemophilia. And I think we're getting there.
Yeah, this is the question I wanted to ask you about your enthusiasm for gene editing, but clearly your enthusiasm is there. So maybe, just before we conclude, you're working hard in this field. You have a great overview of what has been done, what has been successful, what could be successful. So what, in your view, how will the future of gene therapy in this field of hemophilia look like in the next 5-10 years?
Cedric, that's always a tough question to predict.
I know.
We are scientists, not fortune tellers, and there's always unknown unknowns and unknown unknowns to consider. But I think we have a modus operandi in place. You as a clinician and me as a scientist, we do things in an evidence-based manner. We propose hypotheses, we test them, and this is going to give us the answers that we need in order to address the unknown unknowns and the known unknowns. And that has been our modus operandi for centuries, since the Renaissance. So I think we surely have to continue to use the same paradigm to predict the future. But I'd like to paraphrase Abraham Lincoln, who once said that the best way to predict the future is to create it. And I think as a collective here, working on hemophilia, as clinicians, researchers, representatives from industry and so forth, we all have to come together and combine our energy to actually move this field forward. And we can make a difference. We have seen it with, for example, the implementation of the Padua. It makes all the difference in making a better vector, a safer vector, a more efficacious vector. And I think along those lines, we will surely get to safer and more efficacious vectors.
Well, thank you. Well, Thierry, that was great. I'd like to thank you for contributing so actively to this EHAD Academy. And clearly your here learning module is of major interest for all of us. Clearly, gene therapy is a major step in the evolution of new treatments of hemophilia. So I'd like to thank you. And I know how close you are to the field of hemophilia. You are very active within EHAD, attending all of our meetings. I'm sure that if colleagues have questions to you, well, there will plenty of opportunity to meet you again in person at the next EHAT Congress at the beginning of next year. So I'd like to thank you, and I would like to thank our audience for active contribution to this activity. We know that these EHAT podcasts are increasingly popular. They are available on the EHAT platform, but also on other platforms such as Spotify. So don't hesitate to listen to us, sometimes repeatedly, and to advertise for this great activity contributing to spread knowledge and enthusiasm in our field. Thank you, and thank you to the EHAT team for making this kind of activity possible. I wish you a great day and hope to see you soon. Bye-bye.
Thank you very much, Cedric. Thank you very much for having me.